Good morning, dear student. In previous lecture, we already discussed the glycosidic linkage. So, in today's lecture, we are very much interested in the disaccharides. So, what is this disaccharides? What are their different examples of disaccharides and what are their properties? So, let's discuss the first one, that is the definition of these disaccharides. A disaccharides consist of two sugars joined by glycosidic bond. It is called disaccharides. It is a mixture, or it is a composed of two sugar molecules by glycosidic linkage is called disaccharide. In other words, we can say that the disaccharides are composed of two monosaccharides united by glycosidic linkage. There are three common disaccharides such as maltose. First one is maltose, second one is lactose, and third one is sucrose. Again, this maltose, it is a combination of glucose plus glucose. This lactose, it is a combination of glucose plus galactose, and this sucrose is the combination of glucose plus fructose sugar. So let's discuss one by one. The first one is the maltose. So maltose, it is also known as malt sugar. So this maltose, it is not occur in the human body. It hydrolyzes to glucose by the enzyme maltase. Okay, so what is this maltose? It is also known as malt sugar. It is not occur in the body. It is hydrolyzed to glucose by enzyme maltose. Okay, it will shows mutarotation. So we are very much familiar with this mutarotation. Again, it forms an ozone with phenylhydrazine. It reduces the filling solution and benedict solution, and therefore, because why it reduces the filling solution and benedict solution, because it is a reducing sugar. This maltose and lactose is your reducing sugar. So at least you should know this point. This maltose, it is a combination of glucose plus glucose. Again, it is not occur in the body. It is hydrolyzed to glucose by enzyme maltose. It will shows mutarotation. It will react with ozone. It will forms an ozone with phenylhydrazine, and then it reduces the filling solution as well as benedict solution. Its specific rotation is 130 degree. So if you consider these slides, it consists of 2 points, that is maltose. It is comes from the hydrolysis of large polymeric oligosaccharides or polysaccharides such as starch and glycogen, and it is turned hydrolyzed to glucose by maltase. Okay, so maltose, it is a hydrolyzed product of the starch, and we know that starch, it is a polysaccharide or it is an oligosaccharide. So whenever starch undergo hydrolysis, it will gives maltose. So this maltose comes from the hydrolysis of large polymeric oligosaccharides such as starch and glycogen, and it results, and it is further results into the formation of glucose by different enzymes such as maltase. If you see this structure, this maltose, it is consist of alpha D glucopyranose and alpha D glucopyranose. So both the sugar, it is your glucose sugar, it is your alpha D-glucopyranose. Alpha means, if you see the position of carbon number 1, that is anomeric carbon, that, and if you see the CH2OH group of the fifth carbon, they are anti to each other, and therefore they are considered as alpha isomers. And the D means what? It is a member of D series, because the OH group, okay, if you see the OH group in open chain form, it is on right hand side, therefore it is a member of D series. So at least you should know what is this alpha D glucopyranose. So you should know the difference between alpha and beta isomers of this carbohydrates or glucose. So in this alpha D glucopyranose, this OH group of anomeric carbon and CH2OH group of carbon number 5th, it is anti to each other, and they are considered as a alpha isomer. So there is a linkage between carbon number 1 of one isomer to the carbon number 4 of another isomer with elimination of water molecule. and we get a glycosidic linkage, that is the linkage through oxygen, O linkage, okay, the glycosidic linkage, and which results into the formation of maltose, okay. So it is a combination of alpha D glucose and alpha D glucose. If you consider this maltose it is considered as a reducing sugar because it has a free hydroxyl group at carbon number 1, or it will convert easily into its free form. We can say that it is convert easily into its open chain form with free aldehyde group, and therefore this maltose is considered as a reducing sugar. Again, the second important point is that sucrase, lactase and maltase are located in the outer surfaces of epithelial cells lining in the small intestines. So at least you should remember the structure and the different properties which shows by the maltose sugar. So it is not occur in the body, it hydrolyzes to glucose by enzyme maltase, then it will shows meta rotation, it will reduces the filling solution and bending solution because it is act as a reducing sugar, and it will forms ozone with phenylhydrazone, okay. Let us discuss the second one, that is the cellobios, okay. So what is the difference between this maltose and this cellobios? Cellobios is an isomer of maltose. So this maltose and these cellobios, they are the isomers of each other. But what is the difference? If you consider the difference, the cellobios is an isomer of maltose. It differ from maltose only in having the beta configuration at the anomeric carbon, that is carbon number 1 of the left glucose unit. So if we see the difference, that is whatever the hydroxyl group and whatever the CH2OH groups are cis to each other, therefore it is a beta isomers. So it differs from maltose only in having the beta configuration. So maltose having a alpha D-glucopyranose, alpha D-glucopyranose, two molecules combined together to give maltose, while the cellobios, it is beta forms of this glucose, that is beta D-glucose and beta D-glucose combines to give cellobios. So only the difference is that the position of the hydroxyl group at C1, that is anomeric carbon. Otherwise, all the structural features are identical, including a link from C1 of the left unit to the hydroxyl group at C4 in the right hand unit. If you see the diagram, you are very much clear what is the difference between this maltose and this cellobios. So both contain glucose as a monosaccharide, glucose as a carbohydrate, but the difference is one is consist of alpha isomers, alpha D-glucose, and second is considered as beta D-glucose. Let us discuss the next one, that is the lactose. So this lactose, it is the disaccharide of milk, okay. It is present in milk. So this lactose, it is present in milk, and therefore it is also called as milk sugar. Again, it is composed of, or it is consist of a galactose joined to the glucose by beta 1,4 glycosidic linkage. That is how this lactose is formed. So it is formed by the two carbohydrates, one is beta D galactose and another is alpha D glucose. So if you see the difference between this, one of the sugar is your alpha D glucose or alpha D glucopyranose, and the second one is your galactose. So we know that this glucose and galactose, they are epimers of each other. So the second carbohydrate is your beta D galactose or beta D galactopyranose. So it is a 6-member cyclic ring, therefore we can consider it is pyranose structure. So one is alpha D glucopyranose and second is beta D galactopyranose. So lactose, it is present in milk and therefore it is called as milk sugar. It is hydrolyzed to use glucose plus galactose. Again, the same properties such as maltose, it will shows meta rotation. It will forms ozone with phenylhydrazine. Again, its specific rotation is plus 55.2 degree. So only the difference is that in this lactose, one of the isomer or one of the carbohydrate, it is your alpha D glucopyranose, and the second one is beta D galactopyranose. Again, there is a 1,4 linkage, glycosidic linkage is there. So this is regarding to the lactose. Let us discuss the third one, that is sucrose. So if you see carefully, both maltose and the lactose are considered as a reducing sugar, while the sucrose, it is not considered as a reducing sugar. So why it is not considered as a reducing sugar? Okay, so sucrose, what is this sucrose? Sucrose is the common table sugar. It is obtained commercially from sugar cane or sugar beets. The anomeric carbon, so if you see the linkage between maltose, lactose and the sucrose, so there is a difference between linkage. In maltose and lactose, the linkage is 1,4 linkage, while in the sucrose there is a 1,2 linkage of the 1,2 linkage. If you see the linkage, it is 1,2 linkage. So how this linkage? So it is obtained commercially from sugar cane and sugar beet. The anomeric carbon atom of an glucose unit and fructose units are joined in the disaccharides. So you should remember why this sucrose is not act as a reducing sugar, because the anomeric carbon, whatever the hydroxyl group of the anomeric carbon, it is involved in the glycosidic linkage, and therefore free hydroxyl group or free aldehyde group is not available in this structure, and therefore this is considered as a non-reducing sugar. If you consider this fructose, it is mostly observed in 5-membered ring, and therefore it is if you consider, this is your beta D fructofuranose. Okay, one is alpha D glucopyranose, one of the carbohydrate is your alpha D glucopyranose, and the second one, that is your beta D fructofuranose. So pyranose means 6-membered and furanose means 5-membered. So this fructose, it is mostly observed in 5-membered ring, and therefore it is a beta D fructofuranose. So there is a linkage, there is a 1,2 linkage, there is no 1,4 linkage. So anomeric carbon of glucose, it will interact with the carbon number the anomeric carbon of the fructose, and therefore if you see the anomeric carbon atom of a glucose unit and fructose units are joined in the disaccharides. The configuration of this glycosidic linkage is alpha for glucose and beta for fructose. Okay, so the carbon number 1, that is anomeric carbon, and if you consider the second, that is the carbon number 2 of this fructose of 5-membered rings are joined by glycosidic linkage, and there is a 1,2 linkage is there. So the configuration of this glycosidic linkage is alpha for glucose and beta for fructose. Sucrose can be cleaved into its component monosaccharide by the enzyme sucrase. So they hydrolyze to give glucose and fructose by enzyme invertase. It does not exist in the body but occur in cane sugar, pineapple, roots, etc. So these are the examples regarding to this examples of the sucrose sugar. Okay, let us discuss the next one, that is the polysaccharides. So in disaccharide, at least you should know maltose, lactose and sucrose and the difference between these sugars. Okay, in maltose or if you consider the maltose, the example, the malt sugar, that is the combination of alpha D glucose and alpha D glucose, as well as one of the isomer of this maltose is your celobiotes. So celobiotes differ from only the configuration around carbon number 1. So it involves beta isomers of this glucose, and it is considered as a celobiotes. So let us discuss this polysaccharides. What is this polysaccharides? Polysaccharides, or simply glycans, consist of a repeat unit of monosaccharides or their derivatives held together by glycosidic bonds. They are primarily concerned with 2 important functions: structural and storage of energy. Polysaccharides are linear as well as branch polymers. So you should remember the difference. These polysaccharides are linear as well as branch polymers. This is contrast to the structure of proteins and nucleic acid, which are only linear polymers. So this is the difference between proteins, nucleic acids, and polysaccharides, carbohydrates. So these polysaccharides are not only linear but also it is branch polymer. The occurrences of branches in the polysaccharide is due to the fact that the glycosidic linkage can be formed at any one of the hydroxyl group of the monosaccharides. So these polysaccharides are of two types. One is homopolysaccharides and second is one is heteropolysaccharides. So these homopolysaccharides, which is on hydrolysis, gives only a single type of monosaccharides. They are named based on the nature of the monosaccharide unit. Thus, if you consider glucan polymers of glucose, whereas fructones are polymers of fructose. So this glucansen and fructones, they are the polymers of glucose and fructose. So they are considered as a homopolysaccharides. So what is this homopolysaccharide? Which is on hydrolysis to give a single type of monosaccharide, they are considered as a homopolysaccharide. While the second, that is heteropolysaccharide, which on hydrolysis gives a mixture of few monosaccharides or their derivative. So if you see the examples of this polysaccharide, the first one is your starch. So we are very much familiar with this starch. It is a storage polysaccharides of higher plants consisting of two components, that is one is amylose and second is amylopectin, which is present in varying amount. So this starch, if you consider this starch is the carbohydrate reserves of plants, which is the most important dietary source for higher animals, including man. The high content of starch is found in cereals, roots, tubers, vegetable, etc. Starch is an homopolymer composed of D-glucose unit. So you should remember the starch, it is a homopolymer composed of D-glucose unit held by glycosidic linkage, and it is also known as glucosane or glucan. Starch consists of two polysaccharides components. Water soluble, one is water soluble amylose 15 to 20%, and water insoluble amylopectin, that is up to 80 to 85%. If you consider chemically, this amylose, it is a long unbranched chain with 200 to 1000 D-glucose unit joined together by 1,4-glycosidic linkage, and this amylopectin, on other hand, it is a branch chain with 1,6-glycosidic bond at the branching point, and as well as 1,4 linkage everywhere else. Okay, so this is regarding to the starch. The second one is dextrins. So in these slides the structure is not given, so I will request you to try to find out the structure of the starch, dextrins, whatever the polysaccharides. So it is not convenient to add the structure. I will try, but I will give the structure, but at least you try to find out the structure of the starch, dextrins, and then whatever the polysaccharide is there. So what is this dextrin? Dextrins are the breakdown product of the starch by the enzyme amylase or dilute acid. Starch is sequentially hydrolyzed through different dextrins and finally to maltose and glucose. The various intermediates are identified by iodine coloration are soluble starch, amylodextrin, erythrodextrin, and acbrodextrins. So whatever the intermediate are formed, they are analyzed qualitatively. So dextrins are the breakdown product of starch by the enzyme amylase or dilute acids, and it is sequentially hydrolyzed through different dextrins and finally to the maltose and glucose. Let us discuss the next one, that is the glycogen. So this glycogen is the carbohydrates reserve. Glycogen is the carbohydrate reserve in animal, hence often referred as animal starch. It is present in high concentration in liver, followed by muscle, brain, etc. So it is a reserve carbohydrate found in liver and muscles of animals and human being. If you consider this glycogen, it is also found in plant that do not possess chlorophylls, for example yeast and fungi. The structure of this glycogen is similar to that of amylopectin with more number of branches Glucose is the repeating unit of glycogen joined together by 1,4 linkage as well as 1,6 linkage. So I will request you to find out the structure, because it is not easy to insert the structure of these glycogens, amylose, amylopectins, whatever the polysaccharide starch into these slides. Okay, let's discuss the next one, that is Cythin. Chitin. Cythin, it is composed of N-acetyl D-glucosamine unit. So cythin, if you consider, it is a derived sugar. It is a derived sugar. Cythin is composed of N-acetyl D-glucosamine unit. So in next lecture we discuss about this derived sugars and their derivatives. Okay. So cythin is composed of N-acetyl D-glucosamine unit held together by 1,4 glycosidic bond. It is structural polysaccharide found in the exoskeleton of some invertebrates, for example, insects. So this is all about the disaccharides and polysaccharides. In next lecture, trying to make we are very much interested in the derived sugars and their significance. This point is very important. So we are trying to make notes on this point, that is the derived sugar and their significances. Thank you.